Hello everyone, my name is Shan and from Peking University. Here's my topic. Fish faunas have become one of the most threatened groups, and effective protection is urgently needed. eDNA method is popular but lacks standards in fish metabarcoding primer selection and sampling strategy. So, we first evaluated fish metabarcoding primers both in silico and in vitro, and designed a shoreline and interior grade sampling method to test for efficiency. Finally, we conducted fish survey in Beijing based on eDNA after method validation. We reviewed recent published papers and got a list of 22 pairs of fish metabarcoding primers belong to 4 gene regions. In silico PCR means using computer to modify amplification. The figure was about the proportion of amplified taxa, and red bars refer to fish. Taxa specificity was different among these primers, and we can see that primers in 12S and Z2B regions were more stable in amplifying fish taxa, and primers in 12S also performed more stable in species coverage and resolution. In vitro PCR means using true eDNA to amplify the sequence. Compared with in silico PCR, the results of in vitro PCR had a higher amplification specificity of fish, and 12S region amplified the most fish MOTUs, so we choose primers in 12S region in further study. To figure out the sampling strategy in lentic water bodies, we selected 3 lakes of different sizes and conducted grid sampling, both in shoreline and interior. The results show that shoreline samples captured more fish diversity, as the light orange bars showed, and species accumulation curves show that shoreline sampling was more efficient in fish MOTU detection, so we choose shoreline sampling in further study. We are now conducting fish survey in Beijing using eDNA method. We had sampled 109 water bodies and got 78 fish MOTUs after NGS sequencing. Compared to historical data, native fish taxa were decreased while non-native species were increased a lot. The taxonomy hit ratio that this 3 order has the most number of fish MOTUs, and these 4 genera were widely distributed in Beijing. The comparisons between water types show that fish diversities of lentic size were higher and more even distributed than latic, and there were some fish taxa showed different preference for lentic and latic water type. The co-occurrence analysis showed some negative relationships between non-native and native fishes, and the invasive species might be a threat to the local fish biodiversity. That's all, thanks for listening.